Hello, it's Dr. Daystorms, and since we didn't get to finish chapter 5 in class, I wanted to go ahead and post a video lecture to go over some of the specialized proteins and their interactions other than, just hemoglobin and myoglobin, so that way you can see how wonderful and awesome all of this is, and how there's such a wide variety of possibilities with protein. First up, we want to discuss and go over those that are involved in the antibody, the immune response. And to begin with, we're just going to discuss a little bit concerning the types of leukocytes that are associated with the immune system. The first class are the macrophages, or some people pronounce it macrophage, which they use phagocytosis to literally chew up, engulf pathogens and bacteria and things like that. And in an earlier rendition of this video, I tried to add in a video segment of a macrophage doing this. However, it didn't come through and record. So on your own, I suggest that you look it up on YouTube because it's really cool. Another instance of a leukocyte are the B lymphocytes, which are derived from the bone marrow, and they're used to produce and secrete antibodies. Then we have the T cells. And T cells come from the thymus gland, and we can have the cytotoxic T cells, and we can have the helper T cells, which interact with the macrophage, and they secrete the cytokines. This is just a generic IgG structure. And so this is the stereotypical look for the antibodies that most people think of because it looks like the Y. But I wanted to point out some of the specifics here. One is you'll see that there are some portions of it that are indicated by a C, and that capital C stands for the constant domain. So there's very little variation between the different immunoglobulins in their C domains. And then we have the V domains. And the V domains do vary. And so this is the part in particular here that's going to where the antibody will bind the antigen. So an antigen is something that's going to elicit an immune response, but the portion of the antigen that's actually recognized by the antibody is called the epitope. So it's an epitope. And this region here, the antigen binding site, is what's going to recognize the epitope. Okay? And you can have something called a monoclonal antibody, or you can also have polyclonal antibodies. Monoclonal antibodies are those that are synthesized from the clones of a specific B cell. Okay, so all of the identical B cells that make and recognize one specific antigen. So those antibodies are going to be homogeneous, whereas a polyclonal antibody come from different B lymphocytes, and so they're going to recognize different epitopes off of that antigen. So they'll recognize different positions off of that protein or whatever that invading entity is. Okay. I also want to point out the fact that the antibody itself is held together by disulfide bonds. Okay, so it's held together by disulfide bonds, and even though it has it's drawn many times as this characteristic Y shape, you have to realize that that's just a two-dimensional representation of a three-dimensional protein. This is what it looks like in 3 dimensions, or it's a little better representation here. And once again, they still have the heavy chain, which I forgot to point that out last time, which is the light chain. We would have our variable regions and our constant regions. It is a glycoprotein, and so therefore there will be bound carbohydrates. And then you're going to have the region that's going to be binding to the epitope itself. So the IgD, and IgE are often usually those monomers. IgM and IgA can be larger complexes or aggregates. And so, for example, IgM can many times be a pentamer. So this is an example of a pentamer. So each monomer, or each Y, is then held together by this J chain down below. IgA can also be dimers and trimers and things like that. I just wanted to point out the fact that there's a complexity to the immune system itself. Now, the immune system is a great example of the induced fit. And so this is just showing the HIV antigen binding to an IgG. And so what they have done here is, in this first crystal structure, this is actually a space-filling diagram, and there is no antigen bound. And to me, it kind of looks like a hand. So you can have like a thumb and a couple of fingers there, and how the fingers are kind of pinched together. Then whenever the small portion of the HIV antigen is bound, you can see how this hand is opened up. It's like it's grasping on to the antigen. And then if you just have the same conformation here, but what they've done is they've just erased this antigen binding, then you can see what it looks like in B. And you can clearly see how open that what I deemed as fingers before, versus whenever it's a closed hand when it's not bound. And I should point out that this interaction is extremely tight. I mean, it borders, it's not covalent. It is not a covalent interaction. However, the KD can be sub-nanomolar. If we're talking about KDs in the orders of 10 to the minus 10 molar. And so they're very, very tight. And because of this, we can actually utilize this for important reasons in the lab and for laboratory techniques. But it's also very important that our antibodies bind to their antigens very tightly for recognition sites in order to have a good immune response. So, just to reiterate this, the antigens are highly specific for their antibodies. So these antibodies recognize these epitopes and combine them on the orders of magnitude of nanomolar to sub-nanomolar concentrations. We've already discussed polyclonal versus monoclonal antibodies. Monoclonal comes from a particular B cell, so it's a clone of B cells, whereas a polyclonal antibody comes from a larger population of B cells that are different, so it's going to recognize different epitopes of the same antigen. It's used for ELISA, which stands for enzyme-linked immunosorbent assay, and it's also used for immunoblot assays. So this is just an example of the way that ELISA would work. Okay, so you have a well, usually, or some type of platform, but usually it's a well, and it's coated with a sample. So there's a whole variety of antigens on this well, and they just stay. Okay, you have to block anything that's unoccupied with just non-specific protein. So a lot of times I'll put in something like albumin, or even use things like milk, just something casing for milk, just something generic that's not going to elicit an immune response or react with your antibodies of choice. So they use this many times for just to test the concentrations of the analyte that's present. Then you incubate with the primary antibody against that specific antigen. So here they're showing this little, how it's incubated to this little, recognize this as little half circle protein. You let that bind. You'll kind of wash it to get rid of any non-specific binding. Then you add in a secondary antibody that recognizes the first antibody. And this secondary antibody has a enzyme that's linked to it. So the way to determine the concentration of the original epitope is based off of how many of the secondary antibodies there are, since they can only bind to the primary antibody, which binds to the epitope itself. So you add in the substrate to this enzyme, and it gives off a product. And you want to use something that gives off a color, so it's colorimetric or spectrophotometric. And then you just measure how much color is present. So the more of the original epitope that's present in the well, the darker the color will be. That's ELISA. So this here is the example of ELISA, and so you'll always want to have a positive and negative control. I don't know how they set this up to know. We would always use the corners. Like one corner would be the negative control, where there's absolutely nothing in there that would elicit response. One corner would be the positive control, where you deliberately put something in there that would elicit a positive response. And then you can actually measure how darkly, how darkly yellow that the sample is, because it's indicative of the concentration in each sample. This is immunoblotting, and it's very, very similar to the complementary assays that uses radioactive or fluorometric nucleic acid probes for like DNA and RNA for gene expression, so that's similar to a southern blot or a northern blot. You run your SDS gel, then you incubate that with the antibody of choice of whatever protein that you're trying to find. And one way to do it is you could just use a radioactive or fluorescent primary antibody and have it then be exposed to film or the spectrophotometer or fluorimeter in order to see it. Another way is similar to ELISA, to where you incubate it with a primary antibody, then you incubate the gel, or you do a blot, I should say. First you want to do a blot to nitrocellulose paper. Then you incubate that. After the primary antibody, you incubate it with a secondary antibody that would be the radioactive or spectroscopic in some way. And here you can see where a single band shows up to show, okay, there is this cross. You can see how pure the sample is between 1, 2, and 3. I would say this is probably general flow through, an initial washer binding, and then this is the final elution for protein precipitation, a protein purification scheme. The next class of proteins I wanted to discuss really are molecular motors, because these are really, really cool. They're transducers, where they take chemical energy to perform physical tasks. And so it's just amazing how these work, and we're going to talk about them in greater detail next semester when we discuss ATP synthesis. So some of the examples include the ATP synthase and the way that it works, because it's just really cool. It's like a little turnstile for the synthesis, a turnstile for protons in order to make ATP. Muscles, which we're going to go into greater detail with myosin, actin, and their components. Of course, the bacteria flagella, chromosomal rearrangement during cell cycle, because whenever the chromosomes, whenever this goes awry and they don't arrange correctly, you can actually have carcinogenesis can occur. And then, of course, various transporters that can transport, molecules from one side of a membrane to the other side, or either from one location in the body to another location. So in muscle contraction, really there are 4 components I'm wanting to know about. One is myosin, and it's 4 polypeptides, similar to the way that, remember hemoglobin had 4 polypeptides, except this one, they are definitely not identical. We have 2 that are considered heavy chains and 2 that are light chains. And what's really cool about myosin is the fact that it's both filamentous and globular. So if you remember, essentially what I'm trying to say is that, there's this part that's filamentous, really long, it looks more like DNA, really, really long rope-like chains, and then there's a part of it that's, globular on the end, like a little head group. It's really cool. Then we have actin, which actin reminds me of a string of pearls. So there's G-actin, which is globular actin, and then there's the F-actin, which is filamentous. Tropomyosin, which when it combines with troponin, binds to the F-actin to block myosin binding site. And then troponin itself is what's sensitive to the calcium, and it's what regulates tropomyosin in order for muscle contraction to occur. Okay, so here, I mean, it's pretty obvious, at least to me, the fact that you can clearly see the long filamentous tail region, and then we have the globular heads. So there's the light chain, these are the heavy chains. So we can see, even in this electron micrograph here, the fact that you get this long chain and then 2 little heads. And then what happens is that the myosin then aggregates to form these really thick filaments. So you can see how the long filamentous portions all kind of come together almost like a rope or something. And then you can see the head groups that are sticking out and protruding out from the filamentous body. And these get quite large. I know it says 325 nanometers, but you have to realize that on the atomic scale, it's actually quite large. Quite long, I should say. So here we go. This is where we see the long actin filaments. There's the myosin tail, and then we see the little head groups and how that they're gonna click in almost like a ratchet into the way that the actin filament works. Okay, this does require ATP. ATP is the chemical energy that's going to be used in order to do this physical task. And so what happens is we have the actin filaments, which, as I mentioned before, kind of look like long strings of pearls. We have the thick myosin filaments and then the protruding head groups. So what happens first, in order for the muscles to contract, is ATP binds. It allows it to dissociate. Once we have ATP bound to it, it dissociates, the head does, from the actin, so it's free. The ATP is bound really tight and it's hydrolyzed, which means that it is broken apart by water in order to form ADP and inorganic phosphate, which, just as a review, remember, inorganic phosphate is often abbreviated like that, inorganic phosphate. Alright, whenever this hydrolysis occurs, it causes a conformational change. And so the myosin head group gets translocated and kind of flips up. Okay? Then the myosin head group is going to attach to the next bead, so to speak, in that chain of pearls. And it's going to release the inorganic phosphate. Okay, so now we're just continuing because we've blown it up to make it larger, so that way we can see what's happening here. But the inorganic phosphate has left. Okay, this release causes the power stroke. The release of the PI, the inorganic phosphate, is what causes the head group then to shift downwards, while it's still attached to that bead, so to speak, on the actin filament. And by doing that, it causes the entire muscle then to be moved, to shift, okay? And ADP gets kicked out, sort of the way, kind of reminds me of the way that certain guns will discharge the spent shell. ADP is sort of like the spent shell of ATP, gets kicked out, okay? Then the entire process just repeats once again in order for the contraction to continue. So you can see that a muscle contraction actually requires a ton of ATP, because each translocation of a myosin head group requires the hydrolysis of one ATP molecule. So this is just the large overview of everything that I just stated before. So then how is myosin regulated by tropomyosin and troponin, and calcium for that matter? Well, what happens here is that, so the way that this regulation works is that in the resting muscle, we have these long tropomyosin filaments that interact with the actin to prevent the myosin from binding. And then we have troponin, and the three components of the troponin that help this interaction to proceed. In particular, we have the subunits I, C, and T, and troponin I is what prevents the myosin from actually binding. And so then troponin C contains the calcium binding site, which is the signal for muscle contraction to occur. And then troponin T, what it does is it's what links the actual complex to the actin itself. Okay? And so what's going to happen here is whenever the nerve send an impulse that a muscle contraction should occur, the sarcoplasmic reticulum will release this calcium, and this influx of calcium then will bind to the troponin C. Troponin C then will undergo conformational change, which will then allow for the tropomyosin to be relaxed, and that will allow for the myosin to have its binding site revealed for it to be able to do its power stroking that we talked about in the previous slides. I hope that this kind of gives a broad overview, and yet at the same time a couple of specific examples of the wide array of possibilities for protein functions and based off of their structures. And so we talked about the complementarity and even the cooperativity that's possible for certain types of binding and the different binding models. We've also discussed in this chapter the tight complementarity that really rivals a covalent interaction, and that's the complementarity that I'm talking about between the epitope and the antibody. And then now we've also talked a little bit about how you can take a chemical signal in order to have it transduced to do physical work by proteins. And we're going to get into greater details and examples of that next semester. So that's something to look forward to because it's really cool. I'll see you in class.